This module contains the pathways for homologous recombination, which are mainly studied in bacteria. As we have discussed previously, that where there are the homologous recombination is very, very essential and crucial. ये बहुत ज़रूरी है for the survival of cell, and there are different pathways by whom, following the whom, the bacteria or the cell, as a matter of fact, perform this recombination. Among this, the most important one is the recombination BDC, homologous recombination in Escherichia coli in the bacteria. And the RecBCD proteins, as a matter of fact, these are the proteins where they bind to the DNA double helix. Remember, the DNA is the central characteristics, central, जिसे कहते हैं central actor in all the homologous recombinations. And the double-stranded DNA, it breaks and uses its DNA helicase activity to unwind the DNA toward the so-called Chi site. Now, while studying these recombinations are different attachment, I would like to emphasize that you should consider the double-stranded DNA like a strand. एक रस्सी की तरह उसको कंसिडर करें, एक रस्सी जिसमें बाल आया वो, और जब आप उसको अनवाइंड करते हैं तो there are different loops, there are different characteristics, there are different shapes which this DNA molecule or वो जो रस्सी है वो assumes करती है. और if you keep that in mind, it will be very interesting and easy to understand the recombination of the pairs. Mostly this has been studied in the Escherichia coli, and on the DNA molecule there are special sites. The one is the Chi sites, which are found on every 5,000 base pairs in Ecoli genome as it is there. Now, as you know, that there is on the DNA, that is the 3' direction and the 5' direction, and the Chi sites, it occurs over there and the breaks the DNA molecule into the two portions, making the ends of the DNA available. Now, double-stranded DNA and the single-stranded exonuclease, which are the enzymes for the DNA, they become active, and as a matter of fact, the exonuclease and single-stranded endonuclease activities, ये enzymes हैं जो कि एक्टिव होती हैं और DNA के जो किनारे या जो इनके जहाँ से कट होता है उसको वो एक्टिवेट करते हैं. RecBCD pathway, it allows to produce a single-stranded tail, which can then be coated by RecA protein. जिस तरह मैंने पहले कहा, if you visualize that this is a strand, एक रस्सी है जिसको where it is cut or unwound and a single-stranded tail that is made. अब क्या होगा कि RecA जो है, that is the enzyme, that is the protein, it allows the tail to invade a double-stranded DNA duplex and search for the region of homology. ये जो प्रोटीन है, ये जहाँ पे homology है, जहाँ पे एक similarity है, this will automatically search that. And this creates a displacement loop, which is called D loop or it is defined the displaced DNA strand. ये बिल्कुल ऐसे ही है कि अगर वो again we come back to the strand or the जो रस्सी है, अगर आप उसे unwound करें, keeping the both ends tight in our hand, there will be a loop, दोनों strand का loop होगा, and then there will be homology between those strands where these enzymes or those RecA proteins, they will sort out. This has been indicated in this cartoon, where you can see that the one strand is upper strand, it is unwound, and then there is a strand जो है D loop, it is formed. It enzyme is over there, and in the below, the red is a D loop or the displacement loop, and those strands, they are joined together, and the enzyme, it is escaping in the yellow form. Now, during this process, what happens? A nick occurs in the D loop DNA, possibly with the aid of RecBCD. RecBCD means that the recombination of the enzymes which are responsible for B, C, and D. And this nick, जब बन जाती है, it allows RecA and SSB, another enzyme, which is a single-stranded binding protein or enzyme, and it creates the new tail. एक नया सिरा जो है, एक नई side जो है, that is formed. Again, an enzyme DNA ligase. Ligases are there in the cell. They are there, and they seal both nicks together. दोनों जो nicks हैं, उनको ligase जो है, वो seal कर देती है, and it creates a holiday junction. The junction जो seal बनता है, उसको we call that a holiday junction. The branch in the holiday junction can migrate in either direction simply by breaking old base pairs and forming new ones in a process which is called branch migration. जब वो holiday junction जो है वो बन गया तो migration जो है it is either direction 3 prime 5 prime simply by breaking the old base pair. जो old base pairs उधर हैं nucleotide के, they are broken and they form the branch migration in this. Now finally, two DNA strands जो जुड़ गए, that must be nicked to resolve each holiday junction into heteroduplexes or recombination proteins. Heteroduplexes are the DNA products. The RecC protein carries out this function for that, and in this diagram, that is simply identified that how those two pairs are being made. Two alternative products that can be produced in this way depending on which strands are nicked by RecC, another enzyme which is responsible for this.